Yeah, we will discuss. Sure. No, no, no. I didn't start it. Okay. Now I will start. Started recording. Mm. Yeah, we'll discuss about chromosomal aberrations. And actually, many are asking about mock tests. I have given most of the like questions related to different topics already in Telegram. But why we don't take mock test like while you are preparing for something? Because at, after finishing the syllabus, you should take the mock test. What happens will, what happens, preparing mock test is not a tough job. Nowadays, we have so many tools and we have advanced AI. You can text, you can just put some copies of question paper, it will text the question. It's not the way. So many other institutions doing the same. They're just copy pasting the question here and there and putting the same question and redundant questions and selling it for money. It's not the good thing. And it will waste of your time. And if it may be helpful for JRF plant science because most of the questions from the books only are from specified subject. But in the case of SRF, syllabus is too large and too dynamic, and maybe the most of the time the examiners prepares the questions from the recent topics and recent publications, older topics with recent publications. Like what are the advances in polyploidy? What is the advances? Last year they were asked about the comparison of AMI and GG plot. Normally we study G interaction, but we not study about at that much level. The questions came from that. Like that, questions will arise from many directions from the same topics based on the recent advancement and the recent publications in very good journals. It's a very dynamic subject like SRF, genetics and plant breeding. Okay, you can take the mock test. Okay, I will prepare some set of questions based on my knowledge and based on my experience. You will get you will prepare only according to that questions only. You will never focus on other topics. That's the problem. Last year I used to take. That's why you see that many of the coaching institutes are very like who are selling mock test or who are doing this one. It's really ridiculous. I was taken concept based Telegram test. See my questions in Telegram, like the way I prepared the questions. I, I will not add anything like redundant. What is this? Who sent like the actually the questions mainly are based on practicality of the topic. For example, association mapping. If I solve the question, you should understand the topic. In that manner, I used to prepare the questions, and it requires a lot of time and patience. If I want to take a mock test, okay, for namesake, I would have taken, I will open any objectives book, I copy paste them, and I can give. It's not a matter of time. And I shuffle it and I can give. But the thing is, when you are attending something, it should be useful and it should be very, very much worthy. And you should, when you're solving a topic, questions of particular topic, you should be able to understand the concepts. Okay, that's why. And why I'm not taking now because I had to call most of it's not mock test. It is just interpretation of your concept. Mock test is like okay, how much your marks you are getting, how much out of total this one. When you have to talk, take the mock test. When you have confident enough if, enough to, that you finished the whole syllabus before exam to get the confidence like how to attend the test, how to manage the time, and for this purpose you have to attend the mock test. For learning the concept you should solve the bits of like VK Prasan BD Singh is there, then continuous learning will be there. Okay, that's why I'm not taking like the mock test or earlier other institute were conducting like simply copy pasting some questions from BD Singh or other sources. I will not make, and it is waste of time also for you and me also. Then when it comes to the chromosomal aberrations topic, like we have discussed about chromosomes, packing, structure, types, how to analyze the chromosome structure by different binding techniques or different molecular techniques like fission gist, and we'll study what we can study by these techniques. These are called as chromosomal aberrations. You know that in the when organism it's there in the environment, definitely will the the environment should have an effect on the genetic material, either in the terms of mutations, like irradiation will cause the mutation, some chemical mutation will cause the mutation, or some during evolution, spontaneously some mutations will be there. Chromosomal slippage or genetic slippage, recombination slippage. In any other means, there will be some changes or some alteration in the chromosome from the normal structure. That is called as your the chromosomal aberrations. There are many times of aberration that we are going to discuss. There will be change in the chromosomal morphology or the change in the chromosomal number from the normal set or normal morphology or normal set of chromosomes. If any deviation from the normal condition, that is regarded as aberration. Then it can also be regarded as large scale mutations that we have discussed the types of mutations in mutation breeding. Normally, we generally deal with point mutations. These are also regarded as mutation in the larger scale in the genome because if something is deleted from some deletion will happen. Entire region will deleted means most of the genes get deleted and the expression will going to lost. This is the having larger change in the phenotype in the organism. That's why it is called as larger scale mutations. There are different types: structural aberration and numerical aberrations. Like for example, we have haploid, diploid, triploid, and this is called as basic chromosome number. With X, 2X, 3X, they are basic chromosome number. This is just haploid chromosome. This is the chromosome number, gametic chromosome number. Okay. Then, what are the different types of structural aberrations are present? Okay. We have different deletion, inversions, duplication, and translocations. Why these structural aberrations will arise? Because of spontaneous mutations due to age also during some organisms, if it is getting aged, then there will be mutations will arises because of irradiation. You know that end replication problem of telomere end replication problem, the chromosome getting shorter when the cell will ages. This is one typical example of structural aberration which is caused by aging of the cells. Then irradiation, we know that so many X-rays, gamma rays, or some any other rays will actually damages your chromosome and causes the mutations. And there are several transposable elements. There are different types of transposable elements are present in organism. For example, typical example called ACDS system of MAGE or some other system like PI transposable elements, P elements, P elements of Drosophila. There are different transposable elements are there, so that they also create this type structural aberrations in the genome. Then different types as we discussed, there are deletions, duplications, inversions, and interchanges. It also regarded as translocations. We will discuss one by one in detail. What is deficiencies? Means, as the name suggests, it's just a loss of segment of a chromosome. For example, if the chromosome is a normal chromosome, if any loss, for example, either see this particular part is missing, the deletion occurs at the telomere end or intercalary region. Anywhere in the in the chromosome, the deletion may happen. Okay. Then deficiency heterozygote, some genes are present in hemizygous condition that we will discuss. For example, if A, small a, capital A, small a, if by unfortunately your A is got lost, A will start expressing in the cell. This expression of the recessive alleles due to deletion of deletion of dominant allele from the cell, it leads to expression of the recessive allele that is regarded as pseudo dominance. Pseudo dominance. It's very important. to know that uh, normally if you don't know this if you not analyze cytologically, you think that this allele will be the dominant allele, but it may not be the dominant because of there is a absence of counterpart because of deletion of this particular dominant allele. It may expresses. We should always think about when you are analyzing cytologically, like if there is any deletion, because of the deletion, your recessive allele expressing, this is regarded as pseudodominance. That's why differentiating dominance and pseudodominance is very important if there is any deletion in the uh, genome. Then there are types of deficiencies. Either it may be the terminal deficiency or it may be intercalary deficiency, like sorry, interstitial deficiency. At the telomeric end, there will be a deficiency, or inside the chromosome, there will be a deficiency. For terminal deficiency, we need only one break. It just at the end of this one, break will happen, it will get eliminated. But to cause interstitial deficiency, we need two breaks. Okay, we need two breaks to eliminate this particular region. But in case of terminal deficiency, we need only one break. Even though this looks very simple, it asked many times in the exam. Okay, then terminal deficiency will survive only when the broken end heals up and gives rise to telomere. Because we know that telomere are the important part which maintains structural integrity. If telomere lost in the deletions, then definitely the chromosome will get eliminated. This type of deficiency when maintained, when you delete it, then telomere get accumulated again, then only it will survive. This is why the terminal deficiencies are rarely occurs. Then, in several cases, like MC Clintock showed that the broken ends remain sticky, so that after replication, two sister chromatids fuse at a broken end and form dicentric chromosomes. Like anybody heard about breakage of bridge and reunion of the bridge. Like it's a classical phenomenon actually given by the MC Clintock. Because of this aberration, there is a formation of dicentric bridge in the cell. And again, it leads to several chromosomal aberration. This cycle is called as chromosomal bridge uh, reunion cycle. Sorry, uh, bridge fusion and recycle mechanism. I think exact word. It's the problem. That wait 1 minute. Yeah. Actually, what will happen during this one? I will explain the diagram so that it will be very clear. Breakage of bridge and reunion of the bridge and formation of other chromosomal aberrations. It's a very important topic, many times asked in the exam. We'll just explain with some diagram so that it will be very clear. Bridge breakage. You can see that. Share all the notes, don't worry. Like, is there typically present in some. See, for example, if the zygote in the nucleus, first what happens, there will be break happens. Like during the cell division, the breaks will happens. What happened? Telomeres of first region. So you can see this one breakages. Then what will happen? The fusion of the broken ends of the each telophase nucleus. See the ends will join and they're forming the bridge. See, we're having two centromere. It is called as dicentric bridge. Means there will be breakage and there will be formation of dicentric this one. Okay? Bridge, break, and fusion. Then following the prophase, again there will be the break and there will be formation of your acentric, dicentric chromosomes. Acentric, dicentric chromosomes. Because you know the bridge has to break. When there is a break will happens. Again there is a formation of dicentric. Either there may be the formation of acentric also. This phenomenon, classical phenomenon, is called as bridge breakage fusion cycle. Bridge breakage fusion cycle. Because of this phenomena, your most of the structural aberrations will occur. Okay. This is the classical phenomena was given by MC Clintock. Okay. Then, um, like there will be a critical example of deficiencies is like pseudo dominance. Then another thing is like what are the phenotypic effects we are going to get as we discussed pseudo dominance means large deficiencies are lethal even for the heterozygous conditions. Then sometimes what will happen because of the hemizygous condition, there will be like expression of the recessive trait that is called as pseudodominance. In the classical example, notched wing phenotype produced by deletion of the X chromosome and deleted segments carries the Y locus for eye color. And actually, please remember this classical examples. They are very, very important in the developmental biology like notch wing mutations, low winged mutations, eye color mutations, white eye color mutations. These are all happens because of the deletion in the some chromosomal region. And the male hemizygous for X chromosome genes are result in the deletion of lethality in males. And there are several classical examples. There is like that. Just I have given the examples. You just refer it. I will share the book also. In when you take the humans, Cry-do-chat syndrome and anti-Mongolism is a classical example of the deletions. Like there is a deletion in the short term of chromosome 5 belonging to the B group. This syndrome is described by Lejeune and Associate in 1963, and anti-Mongolism deletion in the long arm of G group of chromosome in the 46 syndrome was it is also discovered by Lejeune and co-workers in 1964. By preparing for CSI, these things are very, very important actually. The chromosomal manipulations of human developmental. In developmental biology, these notched wing mutations, several other mutations are very, very important to study the developmental biology. But for us purpose, what is deletion? What is the phenotypic effects caused by mutation? What is bridge breakage fusion cycle? Given it, that much only the enough. And what are the different methods of production of deficiencies? Either by irradiations of the pollen or it may be irradiation of seeds, it may cause deficiencies. As I earlier discussed, use of gametocidal chromosomes. And actually, these gametocidal chromosomes were discovered by Endo in 1988. It is actually the genetic system that allows the isolation of virtually unlimited array of deletion stock in common wheat. Like the major source of these gametocidal chromosomes are Agilops cylindrica and Agilops trinucalis. Okay, the genome of CCDD or UECC. Let I will explain in detail of what is endochromosomes. For example, if you take this is the source of endochromosomes, Agilops cylindrica, and this is normal wheat. When you cross this normal wheat and Agilops cylindrica, this is so selfish that it prefers that their chromosomes of Agilops cylindrica only transfer to the progeny and rest the chromosomes from the other counterpart like normal wheat. it is not going to transfer. Even though it transfer, what will happen? It will break the other host chromosomes, other partner chromosomes. That's why we will get the deletions. Means this preferability of the transfer of their chromosomes to the progeny, what will make this agile of cylindrical chromosomes will create the uh, deletion in the chromosomes which are received from the normal bit parent. This is what they are also, that's why they are also called as selfish chromosomes. Okay, and from this and gametocidal chromosome, we can create the deletion stocks. Okay, and deletion stocks are used for to mapping of the genes. Okay, either production of loops, we can map the gene. There are several ways we can use the deletion stocks. Okay, but you should know what is gametocidal chromosomes. They are also called as endochromosomes because endo discovered it. They are also called as selfish chromosomes because they are so selfish that their chromosome only, they think that their chromosome should only pass to the next generation, and they will create breaks in the counterpart chromosomes which are received from the other parent. Then this is about the deficiencies or deletions. You should know what is deletion, what are the types of deletion, and bridge breakage fusion cycle, and what are the phenotypic effects of deletions, major deletions in the humans, and how we can produce the deficiencies by irradiation or spontaneous mutations, or we can another technique called as gametocidal chromosomes. If you know this much is more than enough. Then another structural aberration is called as duplications. What is duplication? What is duplications? Duplication is the condition where the addition of a chromosome part occurs. For example, you having A, B, C, D, E. Then A, B, B, C, D, E. This P is duplicated one time. There are several types of duplication. Either it may be tandem duplication. So this is actually tandem duplication. In adjacent region only, the duplication of the gene happens. Either it may be reverse tandem duplication. For example, A, B, B, A, B, C, D, E is there. A, B, B, C, C, D is there. Just it's actually reverse of the this BB is just reverse to BBCC. Instead of CC, BB will be there. Duplicated part it just reversed. Okay, A BB, then CC BB, and CC means it's normal order. Instead of BBCC, again it's reverse order, the duplication. It is called as reverse tandem duplication. Adjoining position on the same arm in the reverse order. Then another one is displaced somewhere. Like this duplicated will is there in the somewhere in the genome. Okay, that may be homobranchial displaced duplication or heterobranchial displaced duplication. If it is homobranchial, means same arm of the chromosome. If heterobranchial, it is different arm of the chromosomes. Okay, the duplication is on the, for example, the B genome and the Q arm, the duplicated part present in the P arm. That is called as heterobranchial displaced duplication. There is another type of duplication called as transpose duplication. Means duplication on different chromosomes like we see in translocations. Okay, then like you can see in the clearly A, B, C, D, E, F, G, it's a normal chromosome. If it's a tandem duplication, see this BCD, exactly BCD is duplicated here in the same order, BCD and BCD. And reverse tandem duplication, ABCD, BCD is just DCB. Means see the order is reversed. Then duplication of adjacent arm, ABCD, but the counterpart is in different arm, BCD, heterobranchial duplication. Then non-homologous is the different chromosome only at present. Then it is only duplication. And actually duplications play very important role in the evolution. Because if some gene is duplicated, how this duplication will later, there will be some several changes, mutation in the duplication. It may gives to new genes. Okay, this Bono actually given this term like how the evolution of the genes and gene families from the duplications. The typical example of gene duplication is bar I gene of Drosophila. Means there is a reduced number of facets in the Drosophila where eye shape will going to change. Okay, because of increasing the copy of the bar genes. If more copies of the bar gene, smaller will be the eye facet. Like just take the bar eye gene will related to eyesight is very typical example of duplication in Drosophila. There are several duplication. You see, nowadays what gene families we are observing in the humans, gene families, multi-gene families, all they are because of the duplications. Okay, and I have discussed in detail about these things like how evolution occurs in the genome organization chapter. Please refer it. Then another type of structural modifications they are called as inversions. Inversion is nothing but actually is first time identified by H.J. Muller. Then there's certain stocks carried in the crossover reducers. Like in Drosophila, normally everyone is working on Drosophila. Some of the stocks in the Drosophila, they are getting crossover repressors. Then he thought that why this phenomenon is occurring. Then when they are examined these chromosomes, he thought that there is some part of the chromosomes they are inverted. Okay, see this A, B, C, D, E, F. In the another region, F, E, D was present. Because of this inversion, there is a phenotypic changes. Okay, actually, we know that paracentric inversions they are actually involved in crossover. They are actually crossover suppressors. Means they reduces the crossing over percentage, and this phenomenon actually given by Muller and in the study of Drosophila organisms. And we will study that what are the different types of inversions are present. Either the inversion may be two types, pericentric or paracentric inversions. Pericentric inversion means the inverted segment. This inverted segment having centromere. But if it's a paracentric, the inverted segment doesn't have the centromere. This is the only critical difference between the paracentric and paracentric inversions. And we'll study what are the characters of pericentric inversion, what is the characters of paracentric inversion. If it is a pericentric inversions, the inverted segment involves centromere that we have see some, then it alters the karyotype. When it alters the karyotype, for example, if it is undergrow during packaging stage, what will happen? There will be crossing over. See this crossing over. If it is crossing over, there is a formation of different types of the chromosomes. And see, if it is involving a centromere, definitely it is formation of there is a deletion and duplication. Some chromosomes that progeny we are going to get, some they are having deletion and some they are having duplications. It is a typical nature of pericentric inversion. See, the karyotype is going to change because of some deletion and duplication happen. Definitely, there will be change in the karyotype of the genome, the karyotype of that organism chromosome genome. That's why pericentric inversion will alters the karyotype of the cell. Then it production of monocentric chromosomes with deletion and duplication. Please remember this is the typical character of pericentric inversion. But in the characters of paracentric inversion, we get dicentric chromosomes. But in pericentric, there is no formation of dicentric. Only deletion and duplication and normal chromosome and one is inverted chromosome. Four types of chromosomes are produced after there is a single crossing over in the loop of the inverted loop of the inverted chromosomes. Then normally heterozygote paracentric inversion are partially sterile. Many times they will confuse you. If the homozygous paracentric inversion, they are fertile in nature. Many times they will give this type of question to confuse you. If there is an inversion, but they are in homozygous condition, they are fertile. But only the heterozygous paracentric inversion, they are sterile, partially sterile. When it comes to paracentric inversion, you can see that there is a when there is a crossing over happens in the inverted loop, there is a formation of the chromosomes with centric and acentric chromosomes, and there is a formation centric and acentric chromosomes. and it actually reduces the crossing over in the Drosophila. That's why it is regarded as crossover suppressors. I'm not going in detail because these things are not nowadays. Nobody is going to ask about these things. Only should remember the characteristics. If it is a paracentric, what will happen? If it is a pericentric, what will happen? Then what is the definition? Who identified it? And what are the typical mechanisms? Okay, this much is for the inversions. Then we'll move to the another type of chromosomal aberrations called as translocations. And if you want to study more about this, you can study the Ganesh Prasad book of cytogenetics. Very, very good book, and in a simple manner, he explained everything. Okay. And for exam, nobody will going to ask in detail about the cytogenetics part. And if you want to read for ARS mains, it's very important because they will ask, they'll ask for 10 marks, they'll ask inversions. You want to write the answers. This much is not enough. You should, there are several mechanisms for there. Okay, how these loops will create. If there is any single crossing over, what will happen? If there is a two crossing over in the loop, what will happen? If the any crossing over here also crossing over happens, here also crossing over happens. What is will happen? See, that's why there are two courses. In MSc we have cytogenetics course. In PhD we have applied cytogenetics course. In applied cytogenetics we will discuss all these in detail. For examination purpose, this much is more than enough. Okay. Then there are other type. If you want to read, you please prefer the Ganesh Prasad book. Don't prefer PK Gupta. It's very difficult to understand the language. You will get more confusions than the clarity. That's why please refer Ganesh Prasad book for these concepts. Then another very important structural aberration is translocations. What is translocations? Either it is a non-homologous exchange of the chromosomes. If the exchange of chromosomal part in a homologous chromatid, what will happen? It is crossing over. When it is occurs in the non-homologous chromosomes, then it is called as translocations. Then there is a many ways translocation arises. Either it may be spontaneously, either they be by indigenous mutations, or it can be by some interspecific crosses, meaning for wild hybridization. In that type, also translocation may arises, or some genetically controlled like bridge, breakage, fusion cycle. From that also, you will get some deletions or translocation, uh, structural aberrations. What are the different types of translocations? They are simple translocations because just transfer of one part to the other non-homologous chromosomes. It's called as simple translocations. If there is a shift or reciprocal translocations are also happens, means this part is changed here and from this non-homologous chromosome another part is reciprocally changed. That is called as reciprocal translocation. Either it may be simple end or it may be any interstitial region or intercalary region will transfer to another non-homologous chromosome. It is called as intercalary translocations. And if you study the cytology of the translocation heterozygote, there is a formation of cross-shaped configurations. There is a formation of cross-shaped configuration in the Packetin stage of cell cycle, or either the formation of ring shape, either the formation of ring-shaped structure in the M1 generation. Please remember this packaging stage and M1 metaphase 1 stage is very, very at metaphase 1 we'll getting a cyclic structure, but in the ring structure, but in packaging stage, if there is a translocation heterozygote, if it is a translocation homozygote, this cross-shaped structure will not form. This much, this you should know. We'll see. This is the packaging configuration. You can see clearly here. There is a formation of cross-shaped structure. Okay. And when it is possible, when there is a translocation heterozygote, if there is a translocation homozygote, this structure will not going to form. And in the metaphases, we will getting ring-shaped structures. Okay, based on the orientation of the movement of this one, we will getting this type of either it may be alternate, either it may be adjacent, either it may be adjacent to. That we will see. For example, if it is an alternate one and adjacent one, what will happen? Homologous centromere co-orient and move to opposite poles. But in the case of alternate two or adjacent two, The non-homologous centromere will go to single pole, same pole. Okay. Then you can see clearly here how the moment, for example, you take the alternate, okay, opposite. For example, this two will this two will move, this will move to one pole, and this alternate will move another pole. So that you can see this 1, 2, 3, 4, 5, 6, and this one is moving to one pole, and this one is moving to another pole. Means alternate orientation, and that too, the other one, alternate one means homologous centromere will go. If it is alternate two means non-homologous centromere will go to another side. Okay. And another one is adjacent one, where these adjacent homologous centromere will move to one pole, and another adjacent homologous chromosome move to one pole. But another type adjacent two, a non-homologous adjacent will go move to one pole. See, this non-homologous adjacent will move to another pole, so that we will get a different types of gametes. If it is an alternate one and alternate two, we will get completely functional gamete, because there will be the translocations were compensated. Okay. Then in there, but in the both adjacent one and adjacent two, we are getting non-functional gametes. Okay. This is about the different orientation of the your translocation heterozygote in packaging and mitosis stage. Then, what is the breeding behavior of this translocation heterozygote? I mean, the presence of translocation heterozygote is detected in by, like for example, semi-sterility is there in the any set. Okay, we can think that okay, there may be a translocation heterozygote, and but their semi-sterility occurs because of inversion also. Translocation, like heterozygote inversions. Then how do we differentiate? That's why we had to confirm it by observing the meiosis chromosome, like there will be formation of quadrivalent, and there is a formation of cross-shaped structure that we have seen. Then the functional gametes are obtained from alternate disjunction or two types, like with interchanged chromosomes or with normal chromosomes. Then the functional gametes will give rise to three kinds of progeny, namely translocation heterozygote and translocation homozygote in 1 is to 2 is to 1 ratio. For example, this alternate gamete will fuse, for example, this alternate gamete is selfed. Then we will getting 1 is to 2 is to 1 ratio of translocation homozygote and translocation heterozygote. 1 is translocation homozygote and 2 is translocation heterozygote, and 1 is normal chromosome. This 1 is to 2 is to 1 segregation pattern we are going to get when this functional alternate translocated gamete will selfed and the progenies are produced. Then see, typically we'll see this example. For example, this alternate disjunction happens. We are received the translocation heterozygotes of alternate disjunction. When you mate it, when you self it, there will be formation of one normal, two translocation heterozygous, and one translocation homozygous. This is the typical. By this one we can observe that okay, there is a translocation happens and there is a alternate disjunction. But in other case we can't get. If it is adjacent one and adjacent two, they are not fertile, so that we will not able to do this. Okay, we can't observe the breeding behavior. But if it's a translocation occurs in alternate disjunction, then definitely we will get functional gamete. If we self it, it will get 1 is to 2 is to 1 ratio. If it is a translocation homozygote, there is no formation of cross-creation process. It is a normal structures only we are going to get. Then there is another term called as balanced lethal mechanisms. It was first discovered by the Muller in Drosophila, where if the dominant alleles of a two traits is associated with a recessive lethal effect, they are present in the heterozygous repulsion phase. The homozygotes will not survive and permanent hybridity will be maintained. That we will see here. For example, you can see there will be lethal system, maybe. For example, you take any lethal genes were there, lethal alleles will be there. You know that when lethal alleles, when they are in homozygous condition, then definitely you will not going to survive. The particular cell will not, particular organism will not going to survive. And this lethality may be exist in two types. Either may be gametic lethal or it may be zygotic lethal. Gametic lethal means if the particular gametes are produced. For example, here A is not producing, and here B is lethal gamete. Okay, and here you see AA. This whenever the formation of the zygote of AA, they will not going to survive. Whenever the zygote formation of BB, that will not going to survive. This is called as zygotic lethals, and this is called as gametic lethals. But this will only survive when the two lethals are present in the repulsion phase linkage. When two lethals present in the repulsion phase linkage, then only those gametes will going to survive. That is called as balanced lethal mechanism that typically observed in two organisms called as Oinothera lamarckiana, Arethana muricata. This is about structural aberrations, and I have been very speed like crisp because when we are observing my class, my points are very point to the topic, and it's easy to summarize. But you should prefer the books to read about these things. I'm getting when you read the books, you will get more new points, and it's easy to interpret them if you listen my class. You directly go and read the books. You may get some difficulty in reading this. First, listen the book and don't only prefer on my slide because I may miss so many points because of the maintenance of flow and time, everything I need to take care. And please refer Ganesh Prasad book for these to study structural aberrations and all those things. Okay, I'm covering everything from that book and another book is Keynotes on Genetics by Venkata Reddy. You refer that book also. That is more than enough for the this part of for the SRF. Okay, I'm ending the class about chromosomal aberrations, structural aberrations. In next class, we'll discuss about the numerical chromosomal aberrations. It's a little bit lengthiest topic. I need to cover wide hybridization also in this one. Okay, we will see in the next class. Bye.